Installation Guide. Installing the bold line cage incubator is a simple task that will take about 30 minutes. Start the installation by unpacking the components on a clear table. In doing so, please note the presence of visual tags. They provide a useful and intuitive guidance during installation. Inside the boxes, you will find all the necessary tools and a webcam. If you wish to receive support, please contact our technical team to arrange a video conference. Our staff will be glad to guide you step by step during the installation of the incubator. Position the enclosure on a clear table and disassemble the top and front panel. The shape of the enclosure depends on microscope configuration and on the attached equipment. The enclosure is generally supported by 6 posts. Left and right posts may have different lengths. Attached to the posts, you will find the screws needed to fasten the supporting posts to the bottom panel of the enclosure. Position the left and the right block around the microscope, then position the top and front panel. Position the air heater within 1 meter from the microscope and use the gray flexible tubes to connect it to the enclosure. Mind the red and blue color coding. Connect the air heater and the flow rate sensor to the temperature controller. Connect the touchpad to the only RS-232 port of the controller. Connect the room temperature sensor to the controller and attach it to the microscope pillar. Connect the green specimen feedback sensor. The 2 connectors are clearly marked on the rear panel of the temperature controller. Finally, power the temperature controller, the air heater, and the touchpad. The system is switched on and off from the touchpad. The advanced control algorithm keeps sample and enclosure temperature within 0.1 degrees Celsius from set point by stabilizing the temperature and flow rate of the recirculating air in the air heater. To operate the system in sample feedback mode, connect the green cable to the chamber and gently introduce the green specimen feedback sensor in the sensor lid. Introduce 3 milliliters of distilled water in a 35 millimeter Petri dish and cover it with the sensor lid. Make sure that the sensor is touching the bottom of the Petri and is covered with water. Position the Petri with the sensor lid inside the gas chamber next to the sample. If your interchangeable specimen holder has empty positions, cover them with a dummy dish or with the Okolab covers. Best accuracy is achieved if the Petri coupled for the sensor lid is of the same type as the Petri employed for the sample. If you are employing a multi-wall plate, position the green specimen feedback sensor in one of the wells and cover it with water. If you prefer not to employ a sample feedback sensor, select the enclosure feedback operation mode from the touchpad. In this operation mode, feedback to the controller is provided by the enclosure temperature sensor. Connect the gas controller to the temperature controller through the cableless connector. The positioning order of the controllers is not relevant. Connect one of the 2 transparent silicone tubes to the CO2 input push-in connector. The other end of the tube should be connected to the pressure gauge supplied with the controller. Set the pressure gauge at 1 bar. Finally, connect the pressure gauge to your CO2 supply. Connect the second transparent silicone tube to the air input push-in connector. If compressed air is not available, the Oko pump can be conveniently used. The pump is powered by the gas controller and enabled via touchpad. You will find the proper connection cable in the boxes. Attach the gas output filter. Assemble the vibration-free humidity module and fill the glass reservoir with distilled water. The pre-assembled blue tube should be used to connect the gas controller to the vibration-free humidity module. Use the flexible silicone tube to connect the humidity module to the gas chamber. The desired gas concentration and total output flow rate are set via touchpad. Connect the smart box to the top controller by using the cableless connector. Identify the controller connected to the touchpad and connect it to the smart box with the male-male USB cable. Connect the webcam and the Ethernet cable. Open your browser and type in http://smartbox. With Smartbox, you can remotely view and control the system, and you can download temperature and gas concentration data recorded during the experiments.